your children also themed on various foods?
Foods, no. unless you count a flamingo food, which I'm assuming that's something you do. my eldest daughter was one of those big inflatable T-Rexes.
This is why we chose.
That would be the
Good
best
stuff.
chicken leg.
Chicken, yeah, super delicious. And pink, delicious.
I have a friend who said
I'm mortified. Hello, everyone. Welcome until your last week before the exam. Remember, we have an exam next Monday, 404D. I'll talk about a little bit more of that in a second. Before I start, I wanted to share something with you that I got over the weekend. So this is from a past student. And this is her whole email. I did not alter it in any way. She knew that she didn't do very well in the class, but she's actually in medical school. And they're doing immunology and microbiology right now. Yep, she remembers some things. And then here, although we are going to learn about bacteriophage lambda, she sent me another email about 5 minutes after this one saying, hey, actually we had one line on lambda. But it turns out this class was actually useful, in spite of the bullshit I'm teaching you right now, right? So even if you think that you don't need to remember this stuff, and certainly you could forget this stuff after your last exam, but it actually might be useful for other things in your life. Obviously, this was a very pleasant email to receive, and I'm not doing this this is not like a humble brag or anything like that. This is just proof that some things that you learn in your career, in the classroom, as an undergraduate, actually can help you in further education, okay? All right, thank you. Now, to real classroom stuff. You will note, for those of you that have been through the ELC, I've added something called BLDG. This is a manuscript. It's a little old one now, but it describes the utilization of a bacterial two-hybrid system to identify some protein-protein interactions in E. coli. I will ask you to read that paper on your own time and understand that paper. On Friday, we will go over that paper and break down each of the figures presented in the paper, and then we will talk about how those data add to the overall hypothesis of the manuscript. You will be tested on aspects of this paper, and you will see aspects of that paper on your exam for Monday. Therefore, you will have to know it by the time Monday rolls around. Again, your exam is Monday. The exam will mostly, not all, mostly be on bacteriophage lambda genetics. We've done a pretty good job digging down to the molecular level of how bacteriophage lambda maintains an infection in E. coli. So know that as best as you can. Also, this first section on diversity will be on that exam. While we will get to gram-positive discussions, up until now, all you will have to know for your exam is the gram-negative stuff. We will finish that today. So even if we add gram-positive diversity, you do not have to know that for your next exam. You will have to know it, however, for your final exam in December, which actually isn't all that far away. I think it's about a month from now, which is terrifying and wonderful all at the same time. And then again, that paper that we will discuss on Friday. Questions, comments, and or concerns? Okay, the last thing I want to say before I dig back into bacteria is you are, in fact, running out of time to complete any extra credit assignments. If you want to complete extra credit assignments before the end of the semester, this last month is going to close down on you in a hurry. I promise you, as you have to start doing other things for your classes. If you are unable to make microbiology seminars, which are usually on Thursdays from 11 to noon, a number of other departments can also work as long as you have permission from me. Genetics, biochemistry, cell biology. If you can't make any lectures, please let me know. There are other things that I can have you do. However, those things tend to be a little bit more work. Okay. So we left off last time discussing cyanobacteria, and this slide is up here just to point out the fact that cyanobacteria can differentiate in a culture. They can form these spore-like cells, aconites or dome cells, as well as these heterocysts. And the neat thing about these heterocysts, you can again see very clearly that they're different. They actually run a different physiology. Their job is to fix nitrogen and exchange nitrogen for carbon with the neighboring vegetative cells that are undergoing photosynthesis. So heterocysts are unable to make the chlorophylls required for running photosynthesis and instead make this nitrogenase, which helps them fix nitrogen. So I showed you the sort of the diversity of the cyanobacteria, from soil bacteria to marine bacteria. And these organisms are global carbon cyclers. Two genera, Synechococcus and Prochlorococcus, make up about 33% of the global carbon dioxide fixation. So for those organisms that are going to kind of fix and act as a sink for carbon, for all this carbon dioxide out there, these two organisms run 33% of basically the ability on Earth to suck up that carbon dioxide. But they only make up approximately 1% of the photosynthetic biomass in the ocean. So these two organisms, even though they're contributing extremely little to the overall biomass in the ocean, are really important for fixing carbon dioxide on a global scale. Now, they are actually living in the same environments. So you would think that they're directly competing for this carbon dioxide. But one of the interesting things about these organisms is that they have their light harvesting complexes attuned to different wavelengths of light to allow these organisms to exist at different depths in the water column so that they're not precisely competing for the same light source, but they are actually generating energy from two different wavelengths of sunlight. So how are they going to do that? These organisms photosynthesize using the standard Calvin cycle. You do not have to know this cycle. I will not ask you to reproduce the Calvin cycle. However, you will have to know basically how carbon gets into the Calvin cycle, and the Calvin cycle is broken down into three really distinct phases that I'll ask you to know. In this first phase, carbon dioxide enters in this so-called carboxylation phase. So we're going to use carbon dioxide to actually carboxylate something. And this carboxylation phase is driven by a very specific enzyme called Rubisco. This is the same enzyme that plants use to perform oxygenic photosynthesis. So we condense that carbon dioxide with ribulose 1,5-bisphosphate. Okay. That's shown up here. And after we carboxylate that ribulose 1,5-bisphosphate, we generate now 3 phosphoglycerates. So we split that molecule into 3 phosphoglycerates. So you can see here is our 6-glyceraldehyde 3-phosphate. And then we pass that to the reductive phase. So here's our reductive phase. We have to utilize ATP. We utilize reducing power, shown in blue, so this is NADPH. This is how we generate our phosphoglycerates, which can then go on to feed TCA cycle, right, to make biomass. However, in this last phase, we actually have to regenerate the ribulose 5-phosphate. In order to do that, we run this regeneration phase where we use a number of 3C carbon compounds to shuttle carbon back and forth to make this 5-carbon compound, the ribulose. Again, it requires ATP. So this is an energy and reducing power utilizing process. And the only real function is to get carbon into biomass by turning it into these 3 phosphoglycerates that can go enter central metabolism. Now, it turns out that Rubisco as an enzyme sucks. It has a really poor affinity for carbon dioxide. And in order to really get the concentration of carbon dioxide to levels sufficient for the Rubisco enzyme to work, the cells have to concentrate it. And they concentrate it by packaging the Rubisco enzyme and the carbon dioxide into these specialized organelles. You can kind of see these electron micrographs better in B, but in A, you can see that within the bacterium. These proteinaceous compartments, again, encapsulate the Rubisco enzyme, and they help increase the relative concentration of carbon dioxide so that the enzyme can actually work on it. These compartments are called carboxysomes. Okay. So that was what I had for the cyanobacteria. There are some other interesting non-proteobacteria to discuss. Now remember, these still are gram-negative bacteria. So these are the planktomycetes. There are 4 genera within the planktomycetes, and these organisms are really bizarre. We have set sort of these artificial lines when we discuss eukaryotes versus prokaryotes, the things that make one eukaryotic cell different from a prokaryotic cell. And very often, one of the things that we routinely say is that prokaryotes don't compartmentalize, either into organelles or they don't have membrane-bound compartments that encapsulate something like a nucleus. These organisms actually do that. So here is an electron micrograph of a, I don't know which cell this is, but this is a planktomyces. And you can very clearly see here is our outer cell membrane and our inner cell membrane. They're too close to actually see differences here. But inside the cell, we see this other membrane-bound compartment. which is kind of equivalent to a nuclear envelope. So this hazy stuff inside of this compartment is actually the genome. So these organisms, for whatever reason, have decided to encapsulate their genomes in a membrane-bound compartment, making them very different than almost all other bacteria that we've discussed. Also, these organisms do not make peptidoglycan. So their cell walls do not contain peptidoglycan. So again, another one of the things that we've said over and over and over again is that bacteria contain peptidoglycan. Turns out that some of them don't make peptidoglycan. There are always exceptions to the rules that we teach you. These organisms also appear to have intracytoplasmic membranes or other organelles that you might, again, see something more similar in eukaryotic organisms. And perhaps even most interesting, they usually divide by a process called budding, where you actually sort of bud off or kind of inflate one section of the cell that eventually pinches off to form the daughter cell. This is a mechanism of division often seen in like yeast, where you have budding yeast that divides. But again, this is a bacterium that contains a nuclear envelope, like organelle. They don't contain peptidoglycan, and they undergo budding division. They do not utilize, usually, binary fission.
What is the cutoff between being called a bacteria and being called a eukaryote?
What's the cutoff between being called a bacterium and being called a eukaryote? That is a question that's been hotly debated. If you can imagine how exciting that debate must be for like 30 years, right? So people have also, actually what you should do, if you guys want to have a good conversation, ask that question to Barney Whitman. He would love to sit down and have this discussion with you. As long as you make sure you have a couple of hours, I would strongly recommend it. Or I would not strongly recommend it. But yeah, so it's the question. So really what it comes down to is sequencing, right, of the organism and how it's related evolutionary. So 16 S ribosomal RNAs. It's a great question for those who care. So one of these organelles that is often found in the planktomycetes is the so-called anamoxisome. This organelle is a highly specialized organelle there to reduce nitrite. This is actually chemically speaking, a very difficult process. And these organisms can reduce nitrite to nitrogen gas in order to generate a proton motive force that can eventually be used for generating ATP. Now, these anamoxysomes are designed, so here's a membrane that contains basically this lipid bilayer membrane-bound compartment. Now, in much the same way that the carboxysome can sequester and help drive the concentration of carbon dioxide up in certain of the cyanobacteria, in the planktomycetes, this anamoxisome not only is eventually going to concentrate nitrite, NO2, it's also there to keep hydrazine, a toxic intermediate of the reduction process, away from the rest of the cell. So you can see hydrazine is in here. This is in the inside of the anamoxisome. By keeping hydrazine associated or sequestered to the inside of this anamoxisome, you prevent its reacting with other organic molecules and being toxic to the cell. So the enzymes required to perform the nitrite reduction exist in the membrane of the N-amoxisome. So this NO reduction, you generate hydrazine, and then eventually you can make nitrogen gas and the protons, which will eventually be driven down an ATP synthase, which also exists in this membrane, to generate ATP. And for these organisms that can, in fact, reduce nitrite to generate energy, that is a very specialized niche for these organisms. So these organisms can actually thrive in environments that contain nitrite. Very few other organisms can actually do this reaction. Okay. The chlamydia are obligate intracellular pathogens. You're all well aware of and fond of the chlamydia, of course. And like a very good obligate intracellular pathogen, they, at least in the past, could not be grown in the absence of the host, which has made discovery in chlamydia difficult. Recently, or relatively recently in terms of science, there are now new growth conditions for chlamydia where we can grow them in the absence of a host, which has really enabled the genetic discoveries to be made in this organism. Many of you are probably aware of the fact that the chlamydia have a very interesting and dimorphic lifestyle. They drastically change their cell types during their life cycle. They are non-modal, and they only replicate within cytoplasmic membranes of their eukaryotic host cells. They exist in two different forms. Let me get rid of this thing here. Come on, I need you to. They exist in the so-called elementary or reticulate bodies. And again, like a good obligate intracellular pathogen, they have a limited physiology. So here is a cartoon of the standard differentiation cycle in chlamydia. So the elementary body is the infectious type for chlamydia. This is the type of cell that gets released into the environment. It is resistant to phagocytic uptake and therefore can survive in the eukaryotic host when it's been phagocytosed. Upon phagocytosis, the acidification of these compartments then stimulates the change of the elementary body into the reticulate body. All right, that's this sort of this purple circle without the black square in it. Now, this reticulate body is the replicative form of the chlamydia. So this is the organism that can actually replicate. However, this reticulate body is non-infectious. If you took these reticulate bodies out of the cell at this time while they're dividing, extracted them and tried to infect other cells with them, you would be unsuccessful in doing so. So as these organisms replicate and divide inside this membrane-bound organelle, they inhibit host nucleic acid and protein synthesis and utilize host components for energy. As they enter a state of essentially starvation from depleting host compounds, they begin now to switch back to that elementary body. And then eventually lyse the cell to be released into the environment.
Are they
So
replicating in the endosome?
they are replicating in essentially a phagosome. So these different cell types obviously are
produced to match their environments. So these elementary bodies, the reason that they can be the infectious form is because they are almost like a spore. They have a very rigid cell wall. They're adapted to survive outside of the cell. They're not dividing. They're not metabolizing. And that's shown by the fact that, if you compare the amount of RNA and the amount of DNA that's in that cell, they're basically present at a 1-to-1 ratio, which tends to suggest that these organisms are not transcribing. We're not making message, and we're not making proteins. We're just kind of hanging out waiting. Upon conversion to that reticulate body, that cell wall becomes much more fragile, at least more fragile in the elementary body. We're actively we're actively transcribing message now because our RNA exists in a threefold excess over DNA. So that is one of the reasons why we try to take these reticulate bodies out of this host and add them to another uninfected cell. These cells would be killed during the phagocytosis process. Okay. The spirochetes. The spirochetes are, in fact, gram-negative bacteria. There are actually also some important pathogens within the spirochetes, Borrelia, Treponema, Leptospira. Simply by looking at a spirochete, it almost feels like they evolved to be infectious. And indeed, they have evolved to be infectious due to the ability of these organisms to corkscrew through soft tissues like you. So here are some electron micrographs of a sort of a side view of a spirochete, so these spiral-shaped bacteria, and then a cross-section. And in this cross-section, what you can see is there's this really dark, this electron-dense outer envelope. Contained within that outer envelope, you see this circular thing in the middle. That turns out to actually be the cell body. So this is a cell body that's surrounded by an outer sheath. And between the cell body and the outer sheath, you find a number of these, what they have listed here as flagellum, sort of lining the cell. So they exist between this outer envelope or this outer sheath and the cell body. And it's these flagellum that are actually called axial fibrils. They are associated with the cell body, the length of the organism, and wrap around this so-called the protoplasmic cell body. And interestingly, that sheath and the cell body, or the protoplasmic cylinder, they twist when their flagella rotate. But due to the structure of the flagellum and the way they are attached to the protoplasmic cylinder, the sheath rotates in one direction and the cell body rotates in the other direction, which gives the organism the ability to actually corkscrew through stuff, again, like you. And I'm sure there are YouTubes out there or other videos in scientific magazines where you can actually watch GFPs, a green fluorescent protein labeled Borrelia, I think, actually corkscrewing through an infection and entering into your skin in real time, and actually watch it spread from the site of infection. It's actually quite impressive and gross at the same time. Okay, so after the spirochetes, we're going to start talking about gram-positive bacteria, right? So that means through the spirochetes, at least in terms of diversity, that is what you will have to know for your next exam. We are going to continue to talk about some stuff here today, but this stuff will not be on the exam. It will be on your last exam. So in the non-proteobacteria, we talked about the cyanobacteria. They are very close, we believe, to sort of the bacterial root, or the root of sort of the last common ancestor. The planktomycetes are actually compartmentalized, and they lack peptidoglycan, giving us pause when we try to consider what's the difference between a prokaryote and a eukaryote. Okay, so in the gram-positive bacteria, we have artificially divided these guys up into the so-called high G plus C organisms and the low G plus C organisms based upon their G plus C content of their genomes. In the gram-positive, the high G plus C gram-positives are known as the actinobacteria. So this phylum are the actinomycetes. And when we think of actinomycetes, very often we think of these fungal-like bacteria that can differentiate with hyphae and makes exospores. And it's thought that these are a very important environmental adaptation. So these actinobacteria are very often soil bacteria. You have to live out in a relatively rough environment. The actinobacteria are very often known for their ability to produce these so-called secondary metabolites, and have in fact been sort of co-opted into the drug industry to make antibiotics and anti-tumor drugs. Generally speaking, these actinobacteria can differentiate and form aerial mycelia, so these things actually stand up off of the colony. You can kind of see some differentiation here where you have this fuzzy stuff on top of the colonies, and these are those aerial hyphae sticking off into the environment, which actually contain this chain-like structure. These are spores, but these are exospores, which are very different than endospores, which are spores formed inside of a bacterium. So very often, if you've been working in a lab, you see something fuzzy on your plate and it's a contaminant, and you say, oops, I got a fungus, and you toss it. Yeah, you have to toss it, but that's not always a fungus. Sometimes those are bacterial, and that's like streptomyces, for example. It's also a good idea for those of you who are working in labs not to open those plates and take a look at them and poke at them, because then you disrupt these exospores and you shoot them all over the lab, and then your friends 2 benches away come after you. Okay, so here is a sort of a phylogenetic tree of the 14 orders within the actinobacteria. These are so-called high G plus C organisms because they are all more than 50% G plus C content. They are mostly aerobic. They all look very different. They have some important pathogens in there, especially the mycobacteria, but we'll only talk about a few actinobacteria. So the first suborder are these Micrococcinae, or whatever. I'm not going to ask you how to spell or say any of these things. But in this suborder, the best known are these Micrococcus and Arthrobacter. Unlike most other actinomycetes, Micrococcus does not differentiate. And Arthrobacter has an unusual mode of division. So this is so-called snapping division, which many actinobacteria do. But Arthrobacter has long been used as a model for understanding snapping division. And this method of division is promoted through the use of a two-layered cell wall. So these cells, when dividing, so here are the cells, there's one big cell that's going to eventually divide into two at this invagination, or the septum. The plasma membrane of these cells is the innermost solid line. Outside of that plasma membrane, we find two distinct layers of peptidoglycan, an inner layer and an outer layer. Upon division and forming this septum, the cells only add new peptidoglycan to the inner layer. So now we have a situation where we're adding new peptidoglycan to create two new cells or one new cell, but we're not allowing that outer layer to expand. So as we build new material, something eventually is going to give. And that thing is the outer layer. So at some point, the mechanical stress of adding new peptidoglycan only in this inner layer forces a snap in that outer layer. And it's going to happen at either one of the two locations across from that septum. And it's just going to be the weakest one. It's random as to which portion of that cell actually breaks. But upon breakage, you relieve that tension, and now only one side of these two cells are connected to each other, leaving this really diagnostic V-shaped pattern between cells. So you can have these very long chains of these bacteria stuck together, and snapping bacteria that utilize snapping division have this very distinctive sort of back and forth pattern during their division, due to the fact that all these cells are stuck together and then they've sort of broken away from each other, forming these V-like patterns. So another suborder within the actinobacteria, the Corynebacteriinae, there are some really important pathogens we find in here, like Mycobacterium and Corynebacteria and Nocardia. Could be respiratory pathogens. These organisms undergo snapping division too. But mycobacteria are actually pretty unique. They have a cell wall that is very dense with so-called mycolic acids, which makes them very impenetrable to a lot of other organic molecules and antibiotics, which is why you have to treat these organisms for a very long time with antibiotics if you end up getting tuberculosis. This cell wall, or these mycolic acids, are directly attached to the peptidoglycan. I'll show you a cartoon here in a little bit. But they're also required for forming something that's an outer membrane. Right? So this is a mycobacterial outer membrane. So these are gram-positive organisms that actually produce an outer membrane. And they can be the causative agents of tuberculosis and leprosy. So please don't go poke any armadillos out there so you don't get leprosy. But here is a cartoon of the mycobacterial cell wall. The plasma membrane is shown down here, labeled number 5. Here's our peptidoglycan layer in pink, or purple, I guess that is. And then the mycolic acids, these really long carbon chain length, really, really hydrophobic, greasy molecules are sticking off the peptidoglycan, extending out into the environment. And on top of those mycolic acids, in one, is that we have these lipids, which actually form an outer membrane. This can be seen in these electron micrographs, where we're looking at the cell wall structure of Mycobacterium. So in panel B, here's our cytoplasmic membrane on the inside labeled CM. And then outside of that cytoplasmic membrane, we find the MOM, the mycobacterial outer membrane. This is in contrast to a gram-negative bacterium. So here's a typical gram-negative bacterium in panel C. This is E. coli, showing very distinctly the inner membrane and outer membrane. And you can see how much further apart these membranes are in the mycobacteria. So not only are we dealing with this peptidoglycan layer that's 10 times thicker in a gram-positive when compared to a gram-negative, that outer membrane also exists on top of these really long glycolic acids. So even though we've got two membranes, they are much further apart in Mycobacterium. Mycobacterium leprae is, again, an obligately intracellular pathogen. We can't apparently culture it yet. Anybody who works on a mycobacterium in a lab is crazy due to the fact that they have a multi-day doubling time, at least for the pathogens. For example, Mycobacterium leprae will double in approximately 14 days. Mycobacterium leprae, when you compare it to Mycobacterium tuberculosis, they're obviously closely related, except if you look at their genome sizes, it looks like Mycobacterium leprae lost a megabase of its genome. So it really has evolved to be this obligate intracellular pathogen. And when you dig down a little bit further, you see that these genomes are really bizarre. These organisms are littered with so-called pseudogenes. These genes they look like genes, they look like they probably encoded for a protein at one time, but there's something about them that's broken. Either there's a frame shift in the gene where we've added or taken away fragments of DNA to change the frame so that you no longer make a functional protein when you translate it. Sometimes you get rid of start codons or stop codons. Sometimes you get rid of promoters and ribosome binding sites. But something has changed to make those genes no longer functional. And even with this genome of about 3.4 megabases, only 49% of that genome actually encodes for functional proteins. Most of the defects that we find are in catabolic pathways. So we lose the ability to have a diverse appetite, right, and have really focused on a sole carbon source, which is host lipids. And we're also defective in recombination pathways. Having defects in recombination pathways is likely what has driven this organism to be unable to repair large segments of its genome, thereby driving the loss of non-essential genes. If it's hanging out in this host all the time and sucking up a very specific carbon source and taking the rest of what it needs from the host, it can begin to lose things that it no longer requires. Okay, before I start talking about the Streptomyces, I think I'm going to stop and let you guys get out of here. So we'll pick up on the Streptomyces on Wednesday. Please remember to read the paper on there. You will be expected to discuss it with me on Friday. Get out of here. Enjoy your day.